We know very well the male gamete sperm and female gamete egg are formed by the respective gonads. Male gonad, testis, female gonad, ovary are making the gametes to bring the effective fertilization to happen, right? Now we are coming for the spermatogenesis process. In spermatogenesis process, what we should supposed to know, male gamete is also called as sperm cells, which is very, very important for the fertilization to initiate. Okay, and then second thing is we can understand the spermatogonia structure is also called as spermatogonium if it is singular. Spermatogonia is plural. This plural spermatogonia is making the primary spermatozoite. Primary spermatozoite after undergoing meiosis process, it makes spermatid. How I got the primary spermatozoite from spermatogonia? Because of mitosis. Because of mitosis. So we told n number of time the mitosis process and meiosis process are a type of cell division. Right? In this picture, even though they're not narrated in the display in school book also, sometimes they will not give any one division. We need to correlate those information along with the particular thing. This mitosis happens in this particular phase, right? Mitosis happens in this particular point. So primary spermatozoite is formed from spermatogonium is singular. Spermatogonia is plural. So meiosis 1 is making the secondary spermatozoite. Meiosis 2 is making the spermatid. Right? So this differentiation process is responsible for bringing the sperm that indirectly denotes this differentiation is also called as maturation process. Differentiation also called as maturation process. By this process, the sperm is formed from spermatid. The spermatid is nothing but an immature sperm, and spermatozoa is nothing but a mature sperm. Spermatozoa. Just you need to add zoa suffix. Okay, suffix spermatozoa. This zoa once it is coming means mature sperm. Spermatid is a immature sperm, right? So be very careful with all these things. Okay, because of the mitosis process and meiosis process, spermatozoa is formed, right? So now we are entering into the important segment in this concept is spermatogenesis and hormones. If we're talking about the gonadotrophin releasing hormone is released by the hypothalamus. This gonadotrophin releasing hormone, if it is influencing the pituitary gland, exclusively the anterior lobe of the pituitary. There is no two pituitary glands, only one pituitary, the anterior segment. For example, for my body, this head is anterior segment, right? Like this for pituitary, the head segment is called anterior segment or anterior lobe. From there, we are getting LH and FSH. For this to come, I need the influence of GnRH of hypothalamus. So GnRH of hypothalamus influences the anterior pituitary so that you're getting LH and FSH, right? This LH and FSH are gonadotrophins. This LH is responsible for influencing the Leydig cells to make androgen, to make the Leydig cells. Okay, Leydig cells are influenced by LH so that you're getting androgen, right? So this Leydig cell androgen is making spermatogenesis process to happen. Spermatogenesis process or sperm synthesis process is happening not in the Leydig cells, it is happening in the spermatogonium that we already told to you in the seminiferous tubule. While narrating seminiferous tubule of the testicular lobules, we already discussed seminiferous tubule, which is having the spermatogonium to make the sperm under the influence of androgen. This is happening in the spermatogenesis process, right? So that we already narrated for you. So the continuation of that is exactly appearing in this particular slide. We know very well the Sertoli cells is mainly responsible for nourishing the sperm cells, right? So that is a message we need to understand, right? So if you're talking about, we already told to you, this is pituitary gland. Okay, in the CBSE board exam, they'll say what is this gland exactly? Pituitary gland, and that too anterior lobe of the pituitary, making the LH and FSH together we are calling as gonadotrophins. If it is influencing the testis, what is exactly happening? The testis makes testosterone. So FSH can't make testosterone production. Only LH can make testosterone production. LH brings testosterone production. Testosterone and FSH together control sperm synthesis process. So be very careful. Again, back I'm repeating, testosterone production is not because of FSH or follicle stimulating hormone. Testosterone production is exclusively because of luteinizing hormone in the Leydig cell of the testis. Be very careful. So we should not use, by seeing the picture, don't go for misappropriate judgment on your own and don't say any wrong information, right? So now we are coming for the gametogenesis process. In the gametogenesis process, what we need to understand basically, the sperm length is 60 micrometer in the sense, the mu m denotes micrometer. This mu m is nothing but a 60 micrometer, okay, 60 micrometer. So instead of saying micrometer, we can say 0.00 millimeter, okay, acceptable. And then second thing is the sperm is having an outer cover called plasma membrane. The plasma membrane is a structure which is the outer cover of the sperm, is the outer cover of the sperm, right? So this sperm is having four major important segment, not only the tail, not only the head, between the neck and middle piece is existing. We know very well the head is having the oval shaped structure, which is making the acrosome. Acrosome structure of the head segment, okay, it makes acrosome filament. Acrosome filament when it is trying to move towards the egg. Okay, this acrosome structure of the head is very, very important for penetration of the sperm into the egg. You know very well the lytic enzyme of the Golgi complex brings the oval fertilization. So ova, the word we should not use for human being. Aquatic animals lays ova. Ova should not be used. Only ovum should be used, which is a single egg. This ovum is a single egg which is fused with the sperm cells with the help of the lytic enzyme. We know very well, okay, acrosome structure makes lytic enzyme. This lytic enzyme is very, very important for digesting the egg. We know very well the neck has both proximal and distal centriole, which is present exactly behind the head, and the axial centriole is attached to the distal centriole of the neck. So there are totally three type of centrioles are there: proximal, distal, and axial. So these three centrioles of the neck is making the sperm flexible mobility because of the flexible structure of the filament. The flexible structure of the filament makes the neck movement to happen more nicely, right? If you're talking about the middle piece, which is made up of axial filament, that is inside, that is completely having a lot of mitochondria and cytoplasm. So the axial filament is having a lot of mitochondria and cytoplasm, right? Okay, like this you should not say. Around the mitochondria and cytoplasm, this axial filament is existing, right? And the as I already told you, the motility, flexibility. Okay, whenever when I'm mobilizing, when I'm moving, I need the flexibility that is given by the filament, whereas the energy is given by mitochondria ATP breakdown. Mitochondria ATP breakdown. So be very careful with all these things. So again, back I'm repeating, the Golgi complex keep in the sequence. Golgi complex makes acrosome. Acrosome releases lytic enzyme. That lytic enzyme we used to call as sperm lysin. Sperm lysin, the word we are saying for the lytic enzyme, which is very, very important for digesting the egg outer layer, so that the sperm will have effective entry into the egg for bringing the zygote, right? So the lytic enzyme is also called sperm lysin, right? If you're going for the NEET, they'll say hyaluronidase enzyme, which is not there in the CBSE textbook. So that is why we are not going behind the CBSE periphery, right? So now we are coming for the next segment is Here in the case of gametogenesis process, what we should supposed to know, the tail is made up of axial filament. The sperm moves inside the fluid medium, right? The axial filament is there in the sperm tail, and this makes the sperm mobility to happen. The fluid medium is contributed by the seminal vesicle secretion, prostate gland secretion, and then the bulbourethral gland secretion. The female genital tract is exactly having a very good tail movement. Okay, in the female genital tract, the tail credential or the tail movement credential um of the sperm in the female genital tract. What do you mean on female genital tract? In the female genital tract, cervical canal is present, vagina is present. Okay, ulva is present. Ulva is the first layer, then after that vagina, then after that cervical canal. Then like this, what we are saying, all these structures as a part of the genital tract, right? So genital tract means what exactly it is made up of. What is this fluid denotes? Okay, this fluid denotes what exactly? Okay, the fluid is coming from the male, that is called semen. The fluid of the female is a secretion of the Bartholin gland that we already narrated for you. In the cervical canal, the Bartholin gland secretions are much more. That makes the sperm mobility to happen. In coitus ejaculation, the male human produces these many number of sperm cells for bringing the optimum fertility. Why? Because here 60% of the sperm should have the normal size and 40% of them exhibits a strong mobility. So if the concerned male having a very good strong mobility, we are calling that male as the potent male. If it is not so, impotent male. For that also we are giving different type of fertilization treatment in such a way the concerned couple will get the proper child development, right? The epididymis structure, which is making the sperm to get stagnated for a while before copulation, like this we are not saying, during copulation, the epididymis releases the sperm, but up to that, the epididymis stagnates the sperm. This sperm is having the validity in terms of lifespan of about 1 day. So once it is getting completed, okay, it is coming as a dead cells along the urine. While urinating, you can see the dead sperm cells in the urine, right? So the seminal plasma is having the sperm cells in it, right? The seminal plasma is having the sperm cells in it, right? The seminal plasma is also called as the seminal vesicle secretions, right? So now we are coming for the next major segment after the gametogenesis process. The oogenesis process is also the egg synthesis process, which is basically mimics the sperm synthesis process, but the size, shape, everything is different. We know very well. But of course, the number of mitasis and number of meiosis which is participating are one in number. One mitasis, one meiosis. So one mitasis and one meiosis is a part of oogenesis process. We know very well. Oogonium is also called as egg mother cell. This egg mother cell or oogonium is participating in the egg synthesis process. So in the egg synthesis process, what is exactly happening here is the egg is synthesized. Once the egg is getting synthesized, we are observing the fertilization process in the fallopian tube, right? This oogonium or egg mother cell is making the primary spermatocyte. Is making the primary spermatocyte, right? Or primary oocyte. Primary oocyte is totally different from primary spermatocyte. In this place, mitasis happens. So because of the mitasis, primary oocyte is getting formed, and this primary oocyte is making the secondary oocyte and primary polar body because of the meiosis 1. Because of the meiosis 2, we are getting secondary polar body. So we know very well this is nothing but ooted. This ooted on maturation becomes ovum. So there is the first phase, second phase, and the third phase. Like this, we are dividing: multiplication phase, growth phase, maturation phase. So when I was like you, I used to put the slashes and the word or letter, and this will make me to understand the symbol, multiplication symbol denotes multiplication phase when the oogonium is getting entry into the mitasis for making primary oocyte. Primary oocyte makes the secondary oocyte and primary polar body in the meiosis 1. We all need to understand the secondary oocyte is not becoming the ooted in one go or in one stitch. This secondary oocyte need to travel to reach the fallopian tube. Exclusively the first segment of the fallopian tube is infundibulum. Through that it is getting entry to the second segment of the fallopian tube is nothing but ampullacemus junction. In the ampullacemus junction, this meiosis 2 is happening for making the conversion of secondary oocyte into immature egg is called ooted, and that is getting slowly who is becoming slowly ooted is becoming slowly into ovum. Is also called as mature egg. Okay, so we should be very careful. So meiosis 2 is not getting completed inside the ovary. Inside the ovary, where it is getting completed? In the fallopian tube. Where in the fallopian tube? In the ampullacemus junction. Like this, we should understand, right? So be very careful with all these things, narration. And now we are coming for the next important segment. In the case of gametogenesis process is nothing but you can see this picture here. In this picture, the oogonium denotes the oogonium. Just 1 second. You can see this. The oogonium denotes the egg mother cell, right? So this oogonium is nothing but the egg mother cell, which is making the primary oocyte, and this is making the secondary oocyte, and this is making the ovum not immediately. So the transition stage we used to call as ooted, that we already told to you in the flow chart for you before 2 minutes back. That same thing is getting repeated with the pictorial information for better understanding. We know very well the first polar body is worthless, and secondary polar body is also worthless. So when the secondary oocyte is going for meiosis 2, along with the ovum, which is formed, secondary polar body is also formed. That we already told you in the flowchart. The secondary polar body and first polar body is worthless. It is not going to do anything for the reproduction to happen more successfully. To any level, this primary polar body and the secondary polar body is not helping, right? So be very careful with all these things. Now we are entering into the next important segment is nothing but in the structure of the ovum. Okay, if you're listening this very seriously, okay, zona pellucida layer is very, very important for making the barrier for the microorganism entry. Only the sperm can interact with the zona pellucida. When the sperm is getting interaction, what is exactly happening here is the zona pellucida is getting a structure called corona radiata. So the corona radiata is a differentiated structure of the zona pellucida. This zona pellucida, once it is becoming corona radiata, not allowing multiple sperm entry. Corona radiata won't allow any sperm to get entry into the egg, right? This corona radiata is formed because of one sperm cells, right? So the cytoplasm of the egg is having mitochondria to make ATP, endoplasmic reticulum to bring the sperm maturation, Golgi complex to bring the protein maturation. So Golgi complex is making the protein maturation to happen. For this, it is supported by endoplasmic reticulum, but endoplasmic reticulum is not a platform for the protein maturation. The platform is Golgi apparatus. For example, I am a neurosurgeon. I am doing the brain surgery. So that is the role of Golgi complex. My anesthetician is helping me to do the surgery. My anesthetician is endoplasmic reticulum, right? And the entire hospital power, energy, okay, food accommodation, everything is given by the hospital management. That management is your mitochondria, right? Like this, we need to understand everything in due time. The nucleus has DNA, which is very, very important for the sperm guidance. In the sense what? The sperm should release its enzyme in this particular instant. In this particular instant, it need to move. In this particular condition, it need to secrete the enzyme for getting entry into the egg. Everything is controlled by the DNA of the sperm. We know very well the polar bodies are worthless, that I already told you a number of times, as like um, antipodals of the plant. Cortical granules is very, very important, which release the enzyme. Okay, here additional sperm to prevent the additional sperm binding that we already told you, right? The cortical granules, okay, to promote the post-fertilization process. Post-fertilization process. So be very careful with all these things. The cortical granules the helps the enzyme production to happen, right? You know very well the receptors after getting interaction with the sperm cells. So who is the receptor? Where you are saying the receptor of the egg. In the egg, receptors are there for the sperm. Uh, Once the sperm is getting fixed to the receptor, the egg is getting modulated with corona radiata. Okay, again back I'm repeating. The zona pellucida is again corona radiata because of the sperm attachment to the zona pellucida. So for receiving the sperm cells, certain welcoming structure is there. For example, I'm coming to your birthday party. Your father is receiving me, welcoming me like this. Your father is a receptor. Let us consider. So I'm the sperm cells, all right? And like this, what you go for understanding where the receptor they are narrating for the egg exclusively, right? So mitochondria DNA is totally different from nuclear DNA. Okay, this mitochondrial DNA is taken exclusively from the mother. Okay, it's taken exclusively from the mother. Okay, and it is not, this mitochondrial DNA is not from the father. Right. This is the message you need to understand. Right. So now, I think so you understood this message of the gametogenesis process.